Alright, how are you doing, folks? This is Mr. Ferrari, and I'm here recording for you a virtual lecture. So hopefully you're watching this now Thursday in class. This would be September 30th. And in this lecture, I want to say that we're going to be talking about this idea of membrane transport. For now, I'm actually skipping what would be kind of the history of our understanding of plasma membrane. We got to generally about glycoproteins or so. I'm going to be skipping from where we stopped with glycoproteins and all of our plasma membrane history to topic 1.4, which is the back side of the second page if you have a physical note copy. So again, we're starting here with topic 1.4, this idea of membrane transport. And at this point in time, we've actually done things with membrane transport. We kind of briefly talked about this on Tuesday, if you were there with our osmolarity lab, but there's this there's a handful of ways that molecules or things can move in and out of the cell. We call them simple diffusion, facilitated diffusion, osmosis, and this is one that should sound familiar from Tuesday, as well as active transport. And active transport itself is actually kind of an umbrella term because we could call simple diffusion, facilitated diffusion, and osmosis all a form of what we call passive transport. So we have passive transport, which is simple and facilitated diffusion, as well as osmosis, and then active transport, and we'll look at examples of active transport specifically later on. We call them passive transport methods because they don't require any cellular energy at all. They don't require any energy. Active transport is called active transport because it does require some sort of energy to power it in some way. And we'll look at why it requires energy once we get there. So let's start with this idea of simple diffusion. And I'm kind of miffed that I won't get to talk about this with you in class, but it's okay because I've got some other examples here. But I think the best way, and I believe I mentioned this to everyone in class then on Tuesday, but the best way to think about diffusion is if you take a bottle or a can of like aerosol deodorant or Febreze or Lysol, something like that, something aerosol, and you spray it only in one area of a room, you can sit on the opposite side of the room, and sure enough, after a amount of time, you'll be able to smell it. Because simple diffusion is the natural tendency for molecules to spread out evenly across their available space. It's kind of like a liquid in a sense, where we know that liquids will take up their given volume. So long as they're given volume, liquids will do the best to take up that volume. Gases do the same thing. They just do it a lot better than liquids do. And we call this passive transport because, like I said, we don't need any actual energy for this to happen. Nothing needs to happen, nothing needs to be input for this to occur. Deodorant is not natively alive, but if you were to take a can of spray deodorant, it will still end up diffusing its way across the room, whether you wanted it to or not. And so that's why if you're in a boys' locker room or a girls' locker room, someone's spraying a whole bunch of perfume and deodorant after a gym, why the entire locker room will smell that way, because diffusion, baby. Additionally, this diffusion is something that we call dynamic, and so we create this idea of a dynamic equilibrium. This means that as many molecules cross one way as the other molecules might cross another. Basically, the rate of loss is equal to the rate of gain. And we can kind of see a little quick image of that here, right? What we see in this image here, we see a diffusing of all the particles on the left side of this box, and we see them kind of making their way slowly and randomly over time to the other side, to the point where now we are here on the far right, and we will call this a dynamic equilibrium because now we've got generally the same concentration or the same amount of stuff per volume on each side, and they will kind of freely flow within each other to the point where this will basically and negligibly not change. Sure, there might be minute differences here or there at any given moment in time, but realistically, none of those changes are statistically significant enough for us to really say that it's not at equilibrium. Here's a better look at that as well. And what we'll look at here is this idea of substances diffusing down what we call a concentration gradient. Concentration gradient is this difference in concentration of a substance from one area to another. So if we were to look at this first image here, I would say that the yellow particles here, and maybe they're orange, we won't talk about it, it's fine, but the yellow particles here have a high concentration gradient on the left side and a low concentration gradient on the right side. But then the pink particles here have a high concentration gradient on the right side, but a low concentration gradient on the left side. And so what will happen is we'll see that these will start to diffuse from high to low concentration. And we saw that here as well, right? We see a high concentration here and a low concentration here. And now over time, this is still high, but some not as high, and this is still low, but not as low. And now we're here at this equal concentration. We can see this happening simultaneously with both the pink and yellow particles at the same exact time. This is simple diffusion down, we call it down a concentration gradient. Particles move from high to low. It's like if you're going to drop something off a mountain, you'll watch it roll from the high point of the mountain all the way down to the low point of the mountain. Particles diffuse from high concentration to low concentration gradient. And we can look at some of this in action here. The first thing I want to show you is this simulation here, this model. And what this is just the University of Colorado's wonderful science department and all of their wonderful tools. And it's just a demonstration of basically those yellow and pink particles, more or less. What we see here down here are the center of mass bars. These basically tell us in general for these blue particles where they are most centered. And so right now they're only centered in this area because they're being divided by this green divider, and our red particles are mostly centered here because again they're being divided by this green divider. And what I want to note right now is that both of these centers of mass are kind of bouncing along the middle of these smaller halves right now because this is the only space that they're able to occupy. And so what I want you to do right now is take 3 seconds or pause this video, whatever you got to do, and just think about and maybe provide a hypothesis or estimate or guess or educated thought, whatever you want to call it. What might happen if I remove this divider now? So I've paused my simulation, so take a little bit of time and maybe come up with some sort of 1 or 2 sentence descriptor. You can write this down or it can just be in your head, that's fine. But some sort of descriptor as to what you think you'll see happen with both the blue and the red particles and how their center of mass bars might reflect that. So now I'm going to hit play here. And what we see is, these particles are actually bouncing off of each other, right? The fact is these are real things in 3D space that when they collide, they'll move. They'll give each other energy. This is just basic inertia, like basic physics here, right? And so what do you notice over time what happened? Well, look at my centers of mass. They've both found themselves in the middle of this much larger box. What this tells me is that we have found ourselves in equilibrium. Instead of having all of my blue particles here and all of my red particles here, I've got some blue particles here and some blue particles here, as well as some red particles here and some red particles here. Enough to the point where their centers of mass are basically in the same exact spot here in the middle. We ended with this, but we started with this. And that's just because these molecules will randomly move in the space that they're given, and they will just end up randomly finding their way towards this equilibrium. So we can see it happening again here as well. Now, yes, we see our red has actually gone further, but based off of natural random tendencies, we will find that this will pretty much always waver within this little area here. It won't really go much further than this, and if it does, it's going to quickly come back and find its way back into this little area here in the middle. The other thing that we can look at here really quickly is, say, if we bump our temperatures up and down, a real quick 500 Kelvin, so 50 Kelvin, the red 50 Kelvin. The red particles are at 50 Kelvin. That is negative 370 Fahrenheit. And 500 Kelvin, which is what the blue particles are, is 440 Fahrenheit. So this is really freaking hot, and this is really freaking cold. You will die, and you will die. Just really hot or really cold. And so I'm going to leave the divider in, and I want you to notice, compare and contrast the movement of the blue particle to the red particle. What do you see? Well, what I see right now is I see that, yes, these are both still moving. They're all moving. But my blue particles, which are really hot, are moving a lot faster than my red particles, which are really, really slow. And so if we remove this divider, what's going to happen is, these particles are still in real 3D space. But what we're going to see happen is that we'll see our blue find itself to equilibrium a whole lot quicker than our red will. Our blue is basically pretty much there in equilibrium. Our red is barely over the midline here. We're barely getting to that spot. Both will find their way to equilibrium at some point, but it's going to take this red a whole lot longer to be there. Oh, they just got a good bump there. If you want to look at another video, this video here can help you show it more in the context of a cell. And if you want to see something else in real world, what you can do is take a drop of food coloring and drop it into a vase of water or something and let it sit for a decent period of time. This was a 40-second video here, and you can see even in this 40 seconds, there's one drop which came here right in the center, ends up finding its way across the entirety of this vase here. And it would be interesting is if you did the same thing, but instead you let it sit for maybe an hour instead of just 40 seconds. And you can see what might happen there. Let's get back to our diffusion here. So we're moving high to low concentration gradients. In the body, we see this actually happening every single time that you breathe, right? When you breathe, what happens is you introduce air into your lungs, and we know we like breathing air in our lungs because our air has this wonderful thing called oxygen, in case you forgot about that one. And so we introduce oxygen into our lungs, into the bronchioles and the alveoli of our lungs. What happens is the blood veins, the capillaries that run through these alveoli, that have CO2 heavy blood cells are able to diffuse the CO2 because, again, gases will just freely move throughout the volume that they're given. And so if the air is coming in here, then these gases will be able to just easily diffuse, going too fast, easily diffuse through these membranes here. And again, these are all just plasma membranes, right? Because these are all just cells of some sort. The gases from these CO2 heavy cells can easily diffuse out and into the alveoli so we can breathe it out, and the oxygen we're breathing in can easily then diffuse into these O2, what would become O2 heavy cells, because here they're O2 light, right? They don't have a lot of oxygen here, but they got a lot of CO2, and now they can pick up that CO2 here. Right, and we can kind of see that again. These blood veins here carry these blood cells, and so in this case X here is probably CO2 and Y here is probably O2, and so these will just freely exchange within themselves at that dynamic equilibrium, that the amount of CO2 that's going to pass into or out of the red blood cells is generally equal to the amount of oxygen that will pass into the red blood cells. And now let's talk about osmosis. Osmosis is great because it's actually just diffusion. However, it's specifically the diffusion of water. It is a fancy term for water diffusion, and why people decided to give water its own fancy form, I couldn't tell you off the top of my head. I'd love to know, and it's probably easily looked up, but off the top of my head, I'm not entirely sure. I just know that osmosis itself is a fancy term for the diffusion of water across a selectively permeable membrane. And this can happen, this selectively permeable membrane is probably very similar to, a cell membrane. And so we can take a look here at this diagram, and this left side of this pipe, of this U pipe, we call hypotonic, and we'll get into that definition, but basically it means that there's not a lot of stuff here. And this right side is hypertonic, which basically means that there is a lot of stuff here. And in this situation, what we see here after time is that the water has, quote, moved, and by quote I mean it really did, it just isn't alive to make that decision, it's just natural tendencies, but the water has moved to the hypertonic from the hypotonic solution, because now these two are going to be of equal concentration. These two are going to be of equal concentration now. You see how the density of our particles here is, not very dense, and it's much more dense here on the right side. But then if we look at the the right U pipe, the density looks pretty even, because that's the water diffusing. This is osmosis occurring. It's water diffusing across this membrane to even out the concentrations. Osmosis is this idea of water diffusing from a region of low solute concentration, hypotonic, to high solute concentration, hypertonic. And again, this is passive transport because water is not alive. This particle could be, I don't know, sugar, like sucrose perhaps. And this selectively impermeable membrane may be something like, I don't know, the membrane of a dead potato, cough, cough. There's no energy being put into this environment to push this water across the membrane. It's just going. It's just going. This is passive transport because this requires zero energy. This is moving technically down water's concentration gradient. Now this is probably going to get a little confusing here because we just said, diffusion is moving from a high concentration to a low concentration. And you're right, it is. Of the stuff that we're talking about though, in simple diffusion, it's high concentration of yellow compared to low concentration of yellow, and high concentration of pink compared to low concentration of pink. And if we look here, I've got technically a higher concentration of water here than I do here. Because I've got the same amount of water on both sides, but I've got less stuff on this left side, this technically, you could call it, means that there's a higher concentration of water on this left side. And so, we just said it'll move down its concentration gradient. So from a high concentration to a low concentration gradient. And so now there's actually more water here on the right side. Yes, I know, I see that. But because these concentrations are now even, because there's an equal density of stuff on both the left and the right, we can technically call this, equilibrium. This is balanced. So again, osmosis is a fancy term for water diffusing to even out a concentration gradient. Water moves again from a low solute concentration to a high solute concentration. And so let's talk about those fancy terms at iso, hypo, and hypertonic. This is this idea of tonicity, and it's the ability of a solution to cause a cell to gain or lose water. When we say something is isotonic, I think about iso as in like basketball or lacrosse. I think about like an iso or a one-on-one situation where it's just the ball carrier and a defender, and so it's an even matchup. It's one-on-one. Isotonic is an even concentration across your membranes. This would be isotonic in this sense. The right side of this U pipe is isotonic. We could maybe say that this is isotonic, but again, tonicity is talking more about water, so let's maybe stay away from that. But I can look at this and say that these are both isotonic. This is maybe one whole isotonic solution. Solute concentration is the same as that inside the cell. So solute concentration, the external, maybe this is the external, is the same concentration as that of inside the cell. Maybe this is inside the cell. Okay. We won't see any water movement across that membrane. You could let this U pipe sit for 15 days, and assuming it was a fully closed environment, this would look the same the entire time. The entire time. Absolutely. So it's this hypertonic, and in general, hyper means more. It's like hypertension, high blood pressure. Hypertonic solutions are solutions that have a high concentration of solute compared to inside the cell. The solute concentration is greater outside than that of inside the cell when the solution is hypertonic. And so if we're going to look here, we can say that if this is, maybe we're looking inside the cell here, and we're saying this is the outside of the cell, right? There's a higher concentration of stuff outside the cell here, if this left side is our cell. And so the water will naturally leave to go towards the hypertonic environment to create that isotonic environment. This hypotonic environment will lose water to the hypertonic environment. Again, hypertonic is when a hypertonic solution is when the solute concentration is greater outside or in the solution than it is inside the cell, and so the cell will lose water to balance that out. The opposite is then our hypotonic. Hypo, again, low, like low blood pressure, hypotension. Hypotonic solution is a solution where its solute concentration is less than that of inside the cell. It's less than that of inside the cell. And so we can get a good look at that here. What that might look like is if we have, let's call this an animal cell. This is just a general red blood cell. In an isotonic solution of an animal cell, this is our dynamic equilibrium. We see water entering and leaving freely at generally the same rate, and so we see happy, healthy, smiley face red blood cells. This is the shape we like. But maybe this cell finds itself into a batch of pure distilled water. And pure distilled water is neat because it has zero stuff in it besides water. It's got zero solute. What will happen is because there's a lot of stuff inside the cell and no stuff outside the cell, water will rush through this selectively permeable membrane to try and even out the concentration gradient. Again, because we're going to add water to this concentration to dilute this, and we're going to add stuff here to hopefully bring the concentration up. And so water will try and even this concentration gradient, but because animal cells are not very strong, because it's just plasma membranes, what we end up seeing is this idea of hydrolysis. And so we see cells sometimes bursting in hypotonic solutions. Compare that to a hypertonic solution. Maybe we took our red blood cell and we dropped it into, 1 molar sucrose, perhaps. And what happens now is we've got a whole bunch of sugar here on the outside compared to the inside the cell. And so again, water wants to, in this case, naturally flow towards the high concentration of solute, wants to flow towards a lot of stuff. And so now what we'll see is the water will leave the cell to try and even out the concentration and dilute the outside, and we see this shriveling occurring. In a plant cell, we actually like things to be in hypotonic solutions because, well, they have this wonderful thing called a cell wall, in case you forgot, and that cell wall needs this turgidness, this turgidity that we call it. And so when there's a lot of water inside, it can fill up this wonderful large central vacuole to keep it nice and rigid, to give it a lot of form. Compare that to an isotonic solution where it's still healthy, it's fine, it's living, but there's not as strong of a form holding here, and so it's got this flaccid term to it. And then there's this idea of being plasmolized, where it's in such a hypertonic solution. It's very similar to an animal cell here, where inside the cell we see this kind of loss of cytoplasm in a sense. It's still holding onto the cell wall, but we see like this starburst of a shape because it's basically losing its cytoplasm, for lack of a better way to put it. Right? We know cytoplasm is mostly water, and so it's flowing out the cell here. And this is going to be very important. It is very important to know the differences between both plants and animals and how those cells might react to different types of solutions. So again, hypotonic solution is when the solution has not a lot of stuff and so water will want to go into the place with a lot of stuff to even that out. Isotonics are even, it's a one-on-one, it's 1-to-1 ratio, it's an even flow. The concentration is the same both inside and outside the cell, and a hypertonic solution is a solution that has a lot of stuff outside the cell than inside the cell, and so we'll see water leave towards the outside of the cell to try and bring the concentration up inside and bring the concentration down on the outside. And this is just taking another look at that. So again, isotonics even across the membrane and both sides of it. Hypertonic, we see water leaving. Hypotonic, we see water entering. Same thing we saw here. We see water leaving here because there's a lot of stuff outside, so water wants to dilute that. Hypotonic says it sees a lot of stuff inside, so water wants to dilute that. And again, we saw this here. The cell membrane basically shrinks when a plant cell gets plasmolysed. This plasmolysis, like cell cytoplasm perhaps, and lysis as in the cutting or breaking or destroying. You can see this in paramecium as well that have a vacuole. Right, here's a vacuole of a paramecium. It lives in this hypertonic pond water environment, so there's a lot of stuff outside in the water, and so the stuff the water from the central vacuole will be released because they have this vacuole as a sort of a pump that will try to balance the water, or osmoregulate it is a definition there. Everything we've looked at before has been this idea of simple diffusion, but now let's look at facilitated diffusion. This is still passive transport because it's not going to use any sort of energy. There's still no energy usage. We're still moving down a concentration gradient, right? We're still going to be moving from a high concentration to a low concentration. Actually, how about I show you here where it makes more sense? From a high concentration to a low concentration. It doesn't exactly happen freely. Maybe this is because it's some sort of ion or some other molecule that membranes don't generally like. Remember that membranes like these small and uncharged. So maybe there are some smaller ions that are charged, but because they're small enough that they can fit through some of these maybe transport proteins. Hey, there's our T car coming back. The T and T car, transport proteins. Those membrane proteins that we learned about, here's where they're going to come into play here. Here's where they're coming into play. There are two types of transport proteins. There are channel proteins and carrier proteins. And they're exactly as they sound. They're exactly as they sound. A channel protein is a protein that basically has like a gate, more or less. And they will open or close in response to a certain stimulus, and that is probably going to relate to then what they're going to be bringing in or leaving. And so a student asked me a question, Mr. Ferrari, if proteins don't like, if there are part of proteins that do and don't like water, because some are hydrophobic and some are hydrophilic, wouldn't this be bad because now water can kind of flow freely? And here's really where that comes into play, is because they can often be open or closed depending on what's going on here. The other type of transport protein is a carrier protein, and what happens with carrier proteins is that they will actually undergo a transformation basically to change shape in order to transport the material. And we can take a look at that here. So here's our carrier protein. What we see here in our carrier protein is it's in this first side. It's open to the environment where it's able to bind to whatever this solute is that it's specifically made for. And when this solute binds to it, this causes a shape shift in the protein that opens it up now to the inside. And now the solute can continue freely down its concentration gradient. Right? We see a lot of solute outside compared to inside, and so this will naturally tend to find its way trying to move this way towards the low concentration gradient. And now we're just using this idea, this helper carrier protein, to help with that. Because again, maybe this is some sort of ion like sodium or calcium or something similar that maybe isn't able to freely move through these hydrophobic tails. Now we've got these proteins here that help with that. Now it's a lot easier. And we see a similar thing happening here. Right? So again, these are maybe solutes, or these are maybe particles like sodium or calcium or something similar that can't freely move, but now they've got this channel protein that acts as literally a channel or a tunnel, more or less, that gives these ions a way to flow through and down their concentration gradient. Again, what we've talked about so far, both with diffusion, with osmosis, and now facilitated diffusion, all three of these are passive transport because none of these have required any sort of cellular energy. None of them have required any sort of cellular energy. Maybe they need a little bit of help, such as something like a carrier protein or a channel protein, sure, but at no point in time is there any sort of energy being applied to these molecules to help facilitate this movement. It's just freely happening. And we will pause this one here because this will give me a good opportunity to break up this idea of passive transport and then what we'll talk about with active transport. So go ahead and take a break for a second if you want. Just take a walk, go grab some water, and then find the link to the next video for active transport.